Thank you for coming today. Thank you so much. And let's start. Can you please briefly introduce yourself and your research?
Yes. My name is Professor Ravindra Gupta. I am a professor in microbiology and infectious diseases in Cambridge University, Department of Medicine. And my research actually is very broad. I started as a virus expert and now have become an expert in cell biology, because when we study pathogens we have to understand the cells and humans. So I became very much interested in stem cells, blood cells, immune cells, and increasingly now stem cells. So that's a snapshot of what we've been working on, and we've been addressing big pandemics such as HIV-1 and the SARS pandemic through our work. And also we, of course, conducted only the second ever HIV cure using stem cells back in 2019.
Thank you so much. As Dr. Mazzoka just introduced that most of the customers come to our clinic, they're seeking for the big number treatment. What do you think about, is it possible to culture stem cells into 1 or even 2 billion? And how about the safety concerns?
Yes, it's an interesting question regarding the dosage of mesenchymal stem cells or the stem cells. Certainly, literature shows that high-dose stem cells have been shown to be safe in the clinic using autologous, in other words, self stem cells that are bone marrow derived in the blood, and we are hoping to generate further data on high-dose stem cells through partnerships with Helene Clinic, for example, who have now treated a large number of patients with high-dose therapy. Of course, lower doses will continue to be used, and we are still finding the threshold for how many stem cells are needed for certain clinical benefits across diseases. So more work is needed on the exact dosing. We should bear in mind that to culture large numbers of cells is technically difficult, and the more cells you try to culture, the more careful you have to be. Of course, when you're culturing or maintaining stem cells, you need to check the size and shape regularly to make sure that you're culturing the right kinds of cells and they're not differentiating, and you need to maintain them in the stem cell state for as long as possible. So these are the challenges, and that is why high-dose therapy is a goal, but as we heard earlier, it is not necessarily the most appropriate for every patient. In order to do large numbers, I think there is a problem of removing cells and moving them onto new plates, and so technologies such as vertical flasks have been developed. I know that the clinic is using some specialized flasks for culture. They're also using their specialized medium, which is fantastic. And I know they're very, very stringent and careful in terms of making sure that what they are culturing are pure stem cell derivatives. So that's a really important piece of information.
Thank you so much. As you said that we do use a multi-layer chamber flask to culture the stem cell to make sure that we can use a less passage progress, go through the whole stem cell culture progress, and also that we keep checking on their shape and their CT markers during as Dr. Mazzoka just introduced, using the MSC system to check and to make sure that what we provide to our customer that are really stem cell and only stem cells. Thank you so much. And the second question, as you mentioned, that stem cells, they do differentiate if you don't take care of it. And what do you think about differentiation? Is there any possibility that stem cell might differentiate into normal cells, and even the worst, maybe they can turn into cancer cell? And is there any concern about that?
Yeah, I think I know people have this concern, but from my point of view, there is no risk of these becoming cancerous, because cancer cells have mutations, and usually many mutations, which make them dysregulated in cell cycle. And of course, stem cells very tightly regulate themselves, and therefore the chances of mutations causing cancer are very low. We know that from work on aging that inflammation is a potentially key driver of mutagenesis or mutations in the DNA, which can lead to cancer. But that is happening in the individual, is not happening in stem cells. And so I think the risks are extremely low for cancer progression, and I think that this should not be a concern at all.
Thank you so much. So the stem cells are told to have the effect that can help the human body to for the inflammation effect. So in that case, if we can say that actually stem cell treatment might help you to sort of like decrease the risk of getting cancer?
Yes, I think that this is certainly an avenue which needs to be researched. I think that stem cell therapies may provide environments that reduce inflammation, particularly via essentially exosomes, for example, which may have anti-inflammatory properties. And in my mind, part of the success of stem cell therapy is in reducing inflammation. And as we get older, we get more inflammation in our bodies because of a number of different mechanisms. And I think this is a key thing to reverse in terms of regenerative medicine.
Thank you so much. And the last question. So Dr. Mazzola just mentioned about the stem cell exosomes. What do you think about stem cell exosomes and its effect on treatment?
I mean, exosomes are very exciting. I can give just an example of how modern medicine is using that science. We all know about mRNA vaccines. They use the technology of surrounding protein with lipids or fats, which we have found in our cells. And exosomes are natural packages which the body makes to send signals. And so humans are already leveraging this knowledge by using different types of lipids, but in the human body we of course have the original version of that. And so exosomes are extremely important, I think, in sending signals around the body and potentially reducing inflammation in different contexts. And we know that exosomes can contain or be modified to contain proteins to do different activities depending on the treatment that we want to give. So the future of exosomes is really exciting. In terms of use alongside stem cell therapy, I think again really important. The advantage of exosomes is, of course, they have fewer proteins which the immune system can detect, and so in other words, they are less likely to be rejected if you give exosomes from one person to an unrelated person. So it opens up the possibility of using exosomes from sources for many different patients without requiring the cells from the same person. Of course, autologous exosomes are probably better for a number of reasons. I think we should appreciate that purifying exosomes is difficult, as you showed in your slides, and many people in the past have just used the fluid around cells without purification, and I think I agree. I like the idea very much of purification. You want to know what your product is, you want to know it's pure, and you want to be able to test it. And of course the clinic here, Helene Clinic, rigorously tests the quality of exosomes through a number of methods in the laboratory. So that is really important. And there's another advantage of exosomes, a little bit like freezing food. You can, to preserve food, you can also freeze exosomes to preserve them for transport, and then they can be used a little bit later on, so they do not need to be used immediately. So that's another great advantage. Of course, we also discussed using stem cell therapy plus exosomes from the same patient. And I think that's quite an exciting avenue, because it may reduce the number of cells you need because they are supplemented by exosomes. And I think there could be some really important synergistic activity there. Again, we would like to see or produce data to support those hopes.
Thank you so much. And that's all the questions. Thank you so much for coming today and making the interview.
Thank you very much.
Thank you.